For my next chapter, I chose to do Chapter 7, Viruses and Prions. So first we'll kind of distinguish a little bit between a virus and a prion. So a virus is an obligate parasitic acellular small particle that is composed of nucleic acids and proteins. Viruses are composed of either DNA or RNA, and they are covered in a protein coat. They cause a wide array of diseases in plants, animals, fungi, protists, and bacteria. And then we have a prion, and this is a small acellular protein that is infectious. Prions are composed of single proteins. They mainly cause brain diseases in humans and animals. There is no genetic material found in a prion. Next, we have features of a virus. And we'll kind of get into it and talk about the size of a virus, the function of it, and the structure. So the typical size of a virus is 20 to 200 nanometers. And that's what viruses are measured in, is nanometers. So a virus is 10 times smaller than a bacteria cell and 100 times smaller than a eukaryotic cell. So an infectious virus is actually called a virion. But the function of a cell is to deliver its genome into the host cell to allow its expression. That is the main purpose. But viruses also form a part of our body's microbiome. So we have viruses and they're kind of good. They kind of help our body assemble a immune response to outside foreign particles. Viruses are also harnessed to treat illnesses and deliver vaccines, and they also help diagnose infections. So we'll talk about the structure of a virus, and a virus is either DNA or RNA. Simple viruses have a single molecule of nucleic acid. Viruses are surrounded by a protein coat, and this is called the capsid. A virus does not contain any nuclei. So next we'll kind of differentiate between an enveloped and non-enveloped virus. A non-enveloped virus is also called a naked virus. So an enveloped virus consists of a lipid bilayer that closely surrounds a shell of a virus-encoded membrane-associated proteins. It also contains a viral genome and core proteins that are wrapped within one or more membrane. A naked virus does not contain an envelope, hence the name non-enveloped. It contains nucleic acid and a protein coat, which is that capsid we talked about before. And when it tries to enter a host cell, it enters it via endocytosis. So viruses are named using the binomial nomenclature. They're split into a family and genus. Families are named with the suffix viridae. Subfamilies have the suffix virinae, and the genera suffix is virus. The prefix may be another Latin word or sigla for example, an abbreviation derived from some initial letters. This is a diagram kind of showing you the multiplication cycle of an animal virus. So we have the attachment, penetration, uncoding, gene expression or replication, assembly, and the release. And we'll talk a little bit more and get more in depth about that on the next few slides. So the first step is the attachment. This is where this virus attaches or binds to the host cell, and interactions between viral proteins and the molecules occur on the surface of that cell. Next, we have penetration step, which is the second step that occurs. This occurs through contraction of the tail sheath. That tail injects the viral genome through the cell wall and membrane, but the phage head and remaining components stay outside of the host cell. Next, we have the third step, encoding. So this is when that capsid is removed or broken down. And because of that, the release of that viral genome is injected into the cell wherever replication and transcription will take place. Fourth step is that gene expression or replication. Some animal viruses do follow the normal flow, but there are different ways for them to transcribe and replicate. There's 3 routes listed here. There's route 1, which is the dsDNA. Route 2 is that ssDNA. And route 3 is the dsRNA. Our fifth step is assembly. And this is where those new viral particles are assembled from the genome copies and viral proteins using the host cell ribosomes. And our final step is the release. So this is the sixth step. And this is where those infectious viruses, the virions, are released from the cell. Some viruses like to exit a cell by means of lysis. And what this means is that they completely burst the host cell. The host cell is no more. It is completely destroyed. Next, we'll talk about the cytopathic effect. So this is when structural changes in host cells are caused by a viral invasion. So those infecting viruses can cause lysis of the host cell. And like before I stated, this is where the cell completely bursts and it is no more. It also changes the cell morphology. For example, it can cause rounding of a cell and it just completely messes the cells up. So we'll talk a little bit about how they cultivate these viruses. A virus can be cultivated within suitable hosts, such as a living cell. So the purpose of this is to isolate that virus and identify them in clinical samples. So if they take and put it on an agar and they put it in the right environment and watch it grow, they can kind of differentiate. Another purpose is to do research on viral structure, replication, genetics, and effects on those host cells. 3 types of tissue culture are the organ culture, the explant culture, and the cell culture. And last, we'll talk about prions and the brain. So prions only affect the brain. Disease actually occurs when normal prion protein becomes an abnormal clump in the brain, causing brain damage. So it's basically an abnormal accumulation of protein in the brain. This can cause memory impairment, personality changes, and difficulty with movement. Prion diseases are rare, progressive, and fatal. Currently, it is a untreatable degenerative disorder of the brain.